Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Nadav Ahituv from University of California in San Francisco on this show. Please let me briefly introduce you to our audience. You got your PhD from Tel Aviv University in 2002. You then moved to California to do a postdoc at the Lawrence Berkeley National Laboratory until 2007. After that, you became professor at the University of California, San Francisco, and became director of the Institute of Human Genetics there in 2023. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
So on my end, when I was 13, I was diagnosed with scoliosis. So basically I have a crooked spine, and I got this very nice present, which was a brace that made me very socially lovable. And that did not help. My scoliosis kept on growing. And when I was 16, I basically had spinal fusion surgery. And both my sisters also have scoliosis. They're younger than me. And so that really got me into genetics and complex genetic disease as a kid. And then, growing up, I knew I wanted to go to academia and then, study this. And when I finished my PhD, I was really excited about the genome coming out at the time, and the fact that we actually don't know 98% of it. So genes only make 2% of the genome, and 98% are sort of non-coding. So I was really intrigued by that. And also I thought a lot of the complex disease could be lying in those regions. And so working on that would be very interesting. And so specifically chose a lab that specializes in non-coding genome. And then when I started my own lab, that was the focus of my lab, is looking for non-coding genome and basically genetic disease. And we started to work on limb malformations, but to go full circle, every time I would go to this birth defects meeting and meet somebody who works on scoliosis and, get a sense of what's happening in the field and so forth. And from all those meetings, it ended up that we put together a grant that we got, like a big grant to work on scoliosis and non-coding sequences. And so now my lab also works sort of on my own genetic disease in a sense, which is nice to do.
That's true, and I can imagine that. So let's come to your science that centers around identifying gene regulatory elements and linking nucleotide variation within them to various phenotypes, including morphological differences between species, drug response, and human disease. I mean, you just alluded to that. I just wrote it down and I wanted to still read it. So during my research for this interview, I found 159 publications with your name on it. 9 of those were preprints. So let's try to maneuver through those. Not all of them, but some. So I want to start with a paper that was published in 2007 and investigates an interesting point. What happens to mice if you delete ultra-conserved regions of the genome? So the question is, what happens when you do, actually?
Yeah, so that was a complete surprise. So Gil Bejarano and David Hausler's lab, when he was a postdoc, so both of them discovered these ultra-conserved elements that are, they defined as at least 200 base pairs in length and 100% identity between human, mouse, and rat. So nothing changed over that evolution. And so you would expect these sequences to be extremely important because they're extremely conserved. And so to follow up on that article, what we set out is basically to delete 4 of them in the mouse genome and see what happens. And we were sure, mice will die, we'll see some sort of a phenotype or so forth. And to our surprise, all 4, including one that's 700 base pairs long, had no effect on the mice, at least, in terms of what we could see through, just general phenotyping. And so that was a big surprise. Though later on, my former lab where I did my postdoc and did this work actually kept on characterizing some of these ultra-conserved elements. And definitely, if you do very intensive phenotypic characterization, for at least for 2 of them, they showed that there are very subtle phenotypes, which you could imagine that over time might have some selective pressure. But still, with this conservation, you'd expect or at least that's what we were expecting going into the experiment, having something that's much more deleterious than what we observed.
You then started to functionally characterize tissue-specific enhancers with a set of papers. You looked at liver enhancers that regulate drug-associated transporters, at coding exons that functions as tissue-specific enhancer of nearby genes, or at tissue-specific enhancers in the DLX56 locus. Maybe we can try to summarize those papers and talk about what you found there and what your motivation was and how you got into that.
Sure. So, our motivation was always to find gene regulatory elements, and a lot of them also could appear due to various sort of treatments, for example, or various things that you do to an organism or to cells. And so, drug response is a great example of that, where if you treat, for example, if you give people drugs, right, they could have, they respond differently based on their genetics. And that could be due to genes and proteins that metabolize the drugs. And there's been a lot of work in pharmacogenomics on that. But it could also be due to regulatory elements that lead to differences in expression of these metabolizing enzymes and other transporters or things that come into contact with drugs. And then with drugs, also a key thing is a lot of these regulatory elements could be turned on by the drug itself. So you won't see the regulatory element, it won't be active until you give the drug, and then it sort of shoots up and turns on and it turns the gene on. And so we wanted to characterize a lot of these regulatory elements that respond to various drugs, and did a lot of genomics with and without the drug to find sequences and regulatory elements that respond to the drugs. So that's sort of what we did with the drug-related regulatory elements, and we focused on the liver since it's sort of one of the main regions that is most important almost in metabolizing a lot of these drugs for these studies. And then in terms of coding exons, so at the time, a lot of these high-throughput genomic assays were coming out to find regulatory elements. So before, people would use evolutionary conservation. So if a sequence is conserved, let's say between human, mouse, and rat, like I mentioned, or a fish or so forth, and it's non-coding, you would think that it's a regulatory element. But we always thought that the exons are conserved just because they're exons and code for protein. But when all the sort of techniques came out, like ChIP-seq and later on ATAC-seq and all of these, we started seeing signals for regulatory elements also within exons. And that was really intriguing for us. Like, could there be sort of a dual role for an exon, that it's both coding for protein, but it's also some type of an enhancer or regulatory element in the genome? And so we actually characterized a lot of these and found that a lot of them are actually also regulatory elements and sort of have these dual roles. They moonlight in a sense as, being both regulatory elements and both coding exons. And that was really intriguing for us.
Do they need to be inactive to act as enhancers?
So some of them could be also active, so both active and inactive, but we think that more is inactive, and it might be a way also of inactivating the transcription. So if regulatory elements loop to their target promoter for the gene, it could be a way, we thought, also of silencing that gene, so it won't be transcribed. Now it's sort of an enhancer of another gene, and it cannot be transcribed due to its sort of three-dimensional structure. And that was really intriguing for us. We haven't followed it up too much. It always depends on people's interest in the lab, but I think it's definitely worth following up in the future.
Yeah, so then I won't ask my next question to that and just move on. Then you did a massively parallel experiment to systematically test the impact of copy number, spacing, combination, and order of transcription factor binding sites to gene expression. Can you talk about this, especially about the order of of transcription factor binding on gene expression?
Yeah. So again, when we started, there weren't any high throughput assays to check for regulatory element activity. You can find them based on ChIP-seq and later on ATAC-seq and all these methods. But all these methods are descriptive. They don't really say if that regulatory element is regulatory element, a functional regulatory element. They say that there's transcription factor binding or there's open chromatin, suggesting that it could be a regulatory element. but it's not a functional assay. And the functional assay that exists were sort of testing sequences one by one, where you put a sequence in front of the reporter gene, and if it's a regulatory element, you'll see green if it's GFP or luciferase or so forth. So those experiments are really limited in terms of their scale. And so about 14 years ago, we started collaborating with Jay Shandur's lab in the University of Washington, who pioneered these massively parallel reporter assays. And the trick that Jay came up with was, in addition to having the sequence and the reporter assays, having a 15 base pair barcode that makes RNA. So now if the sequence is a regulatory element, it will turn on the reporter gene, but it will also give you an RNA of that barcode. And so now you can clone hundreds of thousands of sequences and hundreds of thousands of different barcodes. And by RNA-seq, you can just find whether your sequence is a regulatory element or not. So that really revolutionized the field in terms of functional genomics, where you can now, instead of testing a sequence one by one, currently we can test in the lab over 250,000 sequences in one experiment using this technology and see if sequences are functional or not. And so then, you could ask many big questions. One of them is like what you mentioned, where we have a good understanding of the genetic code, how genes code for proteins, but we really don't understand the regulatory code and how regulatory elements function as regulatory elements. So one of the things that we've done with these massively parallel reporter assays is taking sort of neutral sequences we know are not regulatory elements, and starting sort of like a language, starting to put different words, different transcription factor binding sites into them, and seeing what makes them a regulatory element and what doesn't. And we focused on initially on hepatocytes, on liver, because it has a minimal language. It doesn't have too many letters, so to speak. There's about 18 to 20 very important transcription factor binding sites or transcription factors that are important for liver expression. And so we felt that was an easier place to start. And so a lot of our work on grammar and regulatory code has been in the liver, where we basically take these neutral sequences and put, out of these 18 transcription factors, we play around with them in terms of their order, their spacing, the amount, their orientation, and so forth.
What did you find in terms of, were there some specific transcription factors that you found that were maybe dependent on the order that you put them in? Or, is there something that you could mention?
So we see that order actually for very few transcription factors that was important. It's sort of it has been proposed like a billboard model, where no matter where you put stuff, you see expression, and we saw more of that than others. But their order was important for some, but definitely much less. We do see a lot of combinations, which was very interesting, where you have two different transcription factor binding sites. You see increased sort of activity, compared to just having each one by itself. So there's definitely a lot of crosstalk between them. And we also saw orientation differences that are pretty striking, where if you have the plus strand and the minus strand, and if you see transcription factors on one strand versus the other, they sometimes have a very different effect. And also location, how close they are to the promoter, has a lot of effect also on the expression.
Mm-hmm. Yeah, this is hysterical stuff, right? If you expose like the binding site that will help them dimerize, then it probably will help them, right? If it's in the right direction anyway.
Yeah. And another thing we looked at quite a lot also that I think not enough is being done in my mind in that field is also DNA shape. So the way the DNA shapes are in terms of non-BDNA motifs is also very important, we feel, for actual regulatory element activity. And we also play a lot with that on these sort of synthetic sequences to see an effect. And for example, zDNA we saw has a very big effect on regulatory activity through these assays.
Yeah, very interesting. What can all be done to DNA to make it bend and fold into to the right conformation to make it all work?
Yeah.
So then you looked at, during those times, I have the feeling that also the topological domains were discovered, right? It must have been around the same time. And you also looked whether gene expression and regulation are involved in shaping those topological domains and can be used to classify them. So how did you then, is this also connected to the enhancer kind of story that you're doing, and what did you find there?
Yeah, so there we initially actually focused, as I mentioned, when we started the lab, we focused on limb malformations and looking at a lot of them, or maybe I didn't mention before, but a lot of mutations in limb development. And we had a really weird family from that we got in the end, also both genomic DNA through a collaborator and cell lines from Brazil. That had no hands and no feet. Basically, they're truncated. And in the past, there was a nice article that sort of mapped that region near an enhancer that's 1 million base pairs away from a gene called sonic hedgehog. And so that enhancer is known to regulate sonic hedgehog in the limb and turn on that gene in the limb. And when you knock out the gene, just sonic hedgehog, you have exactly that phenotype and no hands, no feet. Usually kids are born dead. They have, holoprosencephaly. They have cyclopia, one eye, and pretty bad birth defects, including, again, truncated limbs. But, these patients had that enhancer perfectly intact. It was fine. And the gene, when we sequenced it, we did whole genome sequencing for this family, was perfectly fine. But surprisingly, they had a 12 kb deletion next to the enhancer. And that region had 3 CTCF sites and that are facing to the gene in the other side that are 1 million base pairs away. And the thought is that enhancers loop to the promoter to turn on and basically the gene expression. And CTCF is important in that loop. And depending on the orientation, if you have 2 CTCF sites facing each other, that will provide that loop. So what we think is happening in these patients is because they're missing these CTCF sites, CTCF keeps on going, passes the limb enhancer, finds the next CTCF site, and then loops. And that's why you don't get expression of sonic hedgehog in the limb, and you get these truncated limbs. And we did a lot of work there, both via seed technologies and also chromosomal staining to show that that is the case in these patients. And so that was sort of our first foray into sort of 3D structure and disease, and definitely showed that the looping is very important. Now, this is intra-TAD, so this is within the TAD. In terms of TAD boundaries, we've collaborated a lot with Lucia Carbone, who's in OHSU, who basically is very interested in the evolution of TAD boundaries. And she's done a lot of work in terms of characterizing TAD boundaries in various different species, including gibbons, which are fascinating because they have this transposome that leads to a lot of recombinations in their genome between species. So you can look even within gibbons to see which TAD boundaries are conserved and which ones are not. And you would imagine that the ones that are conserved throughout all these species are more important than ones that are not conserved in evolution. And so with Lucia, we've basically characterized some of these and also knocked them out either in mice or in cells. And we're also working now to characterize human-specific boundaries that she found that are unique to humans, but you don't see in other species, which we think are important in sort of organizing chromatin organization and gene regulation within these boundaries.
So how does that, is it that gene expression like shapes the TADs, or that TADs shapes gene expression? Or can it work in both ways?
I think both, because, for example, there's beautiful work from Stefan Mundos in Berlin, where he shows, for example, that if you knock out people who have limb malformations also, again, if you have a deletion of a TAD boundary, now what happens is enhancers that's supposed to go, let's say, to the left, start going to the right also, because the TAD boundary doesn't preclude them from doing so. And now you start getting what is called sort of enhancer hijacking, where another gene starts taking, getting directions from this enhancer and is being expressed in places, time points, or levels that you don't want to. And so we think that the 3D structure is very important, and Stefan's work beautifully showed that. And we think that, within the TAD boundaries, like I mentioned, for these CTCF sites that we saw deleted in these patients with truncated limbs, you see that, within it also, that's really important also. So I think all of them combined work together to basically provide this, I'd say, basic background or sort of structure, for gene regulation to work in 3D.
So the assay to discover that would be high-seq in combination with RNA-seq then, right? To see if the tide is still forming and then see if the gene expression is still there.
Yeah. And then if the gene is known to cause any various different phenotypes, you could assay, like, in Stefan's work, he had families that had all of these limb malformations that had these regions missing. They were nice. And then he followed it up with mice to show that you get similar phenotypes. So you could definitely do that also.
Yeah. So now let's fast forward a bit into 2019. Their paper in Science was published with the title CRISPR-mediated activation of a promoter or enhancer rescues obesity caused by haploinsufficiency. Can you talk about this study and its potential impact?
Yeah, so haploinsufficiency is a big issue in human disease. So and basically, it's a tough word, but it means haplo is half and insufficient, half is not enough. And we have basically two genes, usually from our parents, one from mom, one from dad. And if one copy is complete loss of function, meaning not functional, now we have only one functional copy of a gene. And that leads to about 50% of the RNA being made and then 50% of the protein. And for some genes, surprisingly, this is perfectly fine. But when we went through the literature, we found 660 genes that when you have haploinsufficiency, so one copy is fine, one copy is not, you have disease, and the diseases are devastating. Some of these are very severe epilepsy, cancer, polycystic kidney disease, obesity, and intellectual disability. So quite devastating diseases from this. And then the number is thought to be even higher when people looked at whole genome sequencing data sets like NOMAD or others. There's 3,230 genes that they never see a heterozygous loss of function. So likely the number is, in the 3,000 or more for haploinsufficiency. Some of these could be lethal, and that's why you don't see those genes. But definitely there's a lot of disease due to this. And so what you still have here is one copy of a functional gene. It's just giving you 50%. And so since we work on regulatory elements, we thought what we could do maybe is basically make a regulatory element, force a regulatory element to make much more RNA from that existing normal copy. And that, instead of making 50%, maybe now it will make 100%. And you can rescue all of these 660 diseases basically using this approach. And so we chose, we initially thought what disease to choose for this. And we thought obesity is a really great sort of model and proof of principle because, one, in terms of phenotyping, you just measure the weight of the mouse, so it's very easy. And two, if we just upregulate a little bit, we hope that we can still see something because it's a quantitative phenotype. We're just measuring body weight for this. And so we chose for this essay the top two obesity genes, MC4R and SIM1. And just to put it in perspective, if you sequence the top 1 percentile of the population in terms of BMI, body mass index, 5% of them will have mutations in MC4R, and 2% will have mutations in them in SIM1. So this leads to a lot of obesity in humans, and most of the mutations are haploinsufficient, meaning loss of function of one allele. And so it's a very important problem and a very important, a lot of people are walking around with these mutations, and though this could be potential therapy there. And so at the time, CRISPR came out, and then a new form of CRISPR came out, which is sort of the dead Cas9, where you have two mutations in the Cas9, so that basically CRISPR cannot cut the DNA. But you can use it as sort of a delivery truck to bring things to a specific location in the DNA, and you can connect stuff to it, like activators, transcriptional activators that can force, for example, a gene to make more RNA. And this was a beautiful work done both at UCSF by Jonathan Weissman, Wendell Lin, Stanley Key, Lou Gilbert, and Wendell Lin, and And I'm blanking on one last person, which hopefully I'll forget, and also Charles Gerbash in Duke. And so when we heard about this, even before it came out, we thought that this could be a great approach because it could be used to upregulate the existing gene. And so we decided to focus on, as I mentioned, these two genes, and to focus for one of them, for SIM1, both by targeting a promoter and an enhancer that's 230 kb away, 230,000 base pairs away, to see can we rescue it in both cases. And to make a long story short, it worked beautifully. The mice were basically just normal in terms of their body weight. We did this in two ways. One was a transgenic approach, where basically we could upregulate SIM1 in every tissue. And what we saw is we're actually only upregulating where the promoter is active or where the enhancer is active. And this was a hypothalamus enhancer, suggesting that the regulatory element that we go after provides specificity for your basically therapy, that you can only upregulate there and not anywhere else. And then the second way we did it was AAV injections into the brain, into the hypothalamus, where both these genes, MC4R and SIM1, are expressed. And that also beautifully rescued the phenotype. And for one of these genes, SIM1, we actually took the mice all the way to 9 months of age, and we just injected one time at 4 weeks of age. And we still see at 9 months of age that the gene is still upregulated, CRISPR is still expressing. And their body weight is at 30 grams, just like a normal mice, versus because of their genotype, they actually should be, a high 50s to 60 grams in terms of weight, but they look just like a normal mice, suggesting that this could be a long-term therapy. And so following our publication, there's now over 7 companies now that are trying to get this into the clinic, and we hope this will be beneficial. As we started out for a lot of genetic diseases. As I mentioned, there's at least 660 genes that lead to haploinsufficient disease.
So since a 50% reduction led to this phenotype, did you have to do some titration to get it to exactly 100%, or was this then not so important that it's 90 or 110%?
Yeah, great question. So yeah, we did. So we did titrations to get it sort of all the way to 100% with the AAV injections in terms of titer. But we also got a very great question actually from a reviewer to see how high can we go and what happens when we go too high. Like, does that lead to a phenotype? And a lot of these genes, that's a concern. And because if you have overexpression of them, they can also lead to a phenotype. And so we did this for SIM1, where we went really high, on wild-type mice even. So they expressed at 1, and we did twice as much even virus, but the highest we could get to was 1.8. And so we think in this case, SIM1 is a transcription factor, and it has a negative feedback loop likely that auto-regulates it when it's too high, and that's why we could not get too high. Again, that's a hypothesis. We didn't check that out. We had the same thing recently with another gene that we went after with a similar approach, SCN2A, which is the top gene in terms of autism. It has the most known mutations in autism, and it's a sodium channel. So there, it's not a transcription factor that would sort of auto-regulate it. And there we also did, on wild-type mice, twice as much upregulation. And there in particular, if you have gain-of-function mutations in the protein, in the sodium channel, it leads to very, very severe epilepsy. So we're really concerned that overexpression would lead to that. And again, we did not see any phenotype. We think there, because it's a sodium channel, it needs other subunits and other factors to make it to the membrane. And those are at lower levels. And so even if we overexpress, and we don't get as much of it into the membrane. And so it's sort of a different reason why we're not getting too high levels. And that's very different, for example, from a missense mutation, a mutation in the protein that makes it a gain of function.
Mm. So next, you combined FACS sorting of leptin-responsive hypothalamic neuron nuclei with genomic and epigenomic approaches. And epigenomic approaches means RNA-seq combined with ChIP-seq and ATAC-seq. To generate a comprehensive map of leptin response-specific regulatory elements. So what was the motivation behind this? Was this combined with this obesity story, and what did you find?
Yeah, so the lab, since it started, has always worked on obesity, and the issue with obesity, most of the obesity, the most important region is the hypothalamus in the brain, and there's really not good cell lines or ways to work. The hypothalamus is very complex. And so the best way is to work with mouse models, basically. And as I mentioned before, in terms of drug response, a lot of things also in the hypothalamus, or a lot of regulatory elements, could respond to, in this case, various hormones like leptin. And so what we did was work with mice and basically fast them, and then either give them leptin or not give them leptin, to look at regulatory elements and genes that basically turn on when you give leptin. And then did a lot of both genomics, RNA-seq and ChIP-seq and so forth, to find regulatory elements that respond to leptin specifically in the hypothalamus. And again, this is almost impossible to do in cell lines, just because there's no good cell lines for hypothalamus. You can find them, they exist, but they don't really recapitulate the hypothalamus as much as an organism. And through that, we found a lot of regions that are very interesting in terms of leptin response, and of course would be very interesting also in terms of obesity and other sort of related hypothalamus phenotypes in that work.
So if we go just a tiny bit into the methodologies that you use, so the RNA-seq, the ChIP-seq, and the ATAC-seq, was this done like in a multiomics approach in the same cell, or did you do this as a single cell approach, or how did you do that?
Yeah, so this was sort of pre-single cell revolution, so to speak. So we did it in bulk, but we recently have an article that's still on bioRxiv where we did this in a single cell approach. And this we did both in mouse hypothalamus and in human hypothalamus, in males versus females also, and found a lot of also interesting differences between sexes in the male and female hypothalamus. And in addition, a lot of interesting regulatory elements that are associated with obesity.
So forward to last year, which is 2023, you examined all possible pair and triplet combinations, permutations, and orientations of 18 liver-associated transcription factor binding sites, DFBS, which sounds like a lot of work, but also very interesting to see how this all works together. So why was it 18 transcription factors, and what did you find when you did this experiment?
Yeah, so as I mentioned before, we're always interested in the regulatory code, and the liver sort of provides a good sort of simple language. I mean, you can envision it to Hebrew, for example. It has 22 letters, very simple language, and so versus Chinese that has many characters. And so we felt it's a bit of a simpler start to decipher the language there. And so, as I mentioned before, there's about 18 words or transcription factors in the liver that are very important. And so it's a minimal language that we can play with. And so there we did these massively parallel reporter assays. And over time, we did the similar approach years ago, where we just, the technology of massively parallel reporter assays just started, and we were limited in the amount of sequences that we can test and their length. And here we could do 200,000 sequences versus before we did a few thousand. And we could do the much longer 270 base pairs versus before, I think there were about 160 base pairs. And so one, it increases our number. Two, it increases our real estate in terms of the amount of sequences and length of sequences that we can play with. And so using that, we could really learn, and we learned more about the regulatory grammar through this article, basically learning about combinations, about orientation, about order, and so forth, which I mentioned before. And just the sheer scale of it really provided us a better way to improve our understanding of the code.
Is there a brief summary that you could give to that? Is there a take-home message?
So I think, one, we saw that order isn't as important for most regulatory elements. We saw that usually homotypic, so the number of, so let's say you put the same transcription factor over again, it usually increases the activity, but not in all cases. And we saw that heterotypic, so if you have different transcription factor binding site, that usually gives you an increase in expression, but sometimes they also compete with one another. And we saw that what strand you're on is very important also, but not in all cases. So all of that is to say that there's no golden rule. And some transcription factors may behave in a certain way versus others in terms of their grammar. And just showing how complex things are. I think, a lot of this sort of in terms of solving this also is now coming from AI and machine learning, and a lot of groups are doing that. And we hope that, our data, also our experimental data, will be useful for a lot of those AI tools. And we're generating a lot more of these sort of massively parallel reporter assays in order for having those data sets to train on.
What do you think, or what is the key thing that AI can bring to the table here? Is it that it can more easily detect patterns, or that it's just the sheer amount of data that they can then work with?
So I think on our end, we're trying to just provide the sheer amount of data. I mean, we're also using AI to try and learn. But I think there's beautiful work recently, both from Alex Stark's lab and Stein Art's lab, with flies, where they are using basically AI to learn tissue-specific regulatory elements. And then they use the AI to basically design those elements, and then they test them in flies and see nice expression pattern exactly in the cell types that they wanted or predicted to have. So I think the more sequences we have and the more sort of functional assays we have, the more data sets we'll have to train for AI, the more we'll learn. And I think in particular, these sort of synthetic sequences where everything's controlled, right? And we put words in certain locations really could help AI in terms of the learning, and it provides a really great data set for that. In addition to other sort of large scale data sets. So for example, as part of the ENCODE project, we recently generated 680,000 sequences for MPRA in 3 different cell types. And so that also provides a lot of sequences that could be learned on and trained on. And these are not synthetic sequences. These are sequences that exist in the genome and that could be specific to each one of these 3 cell types. So again, generating a lot of these data sets for people to train on, I think, is really important. And the more we have, of course, the better AI does, right? The more language we have.
And last but not least, early this year, you used an integrative single-cell sequencing and single-cell ATAC-seq experiment on insectivorous and frugivorous bat kidneys and pancreases and identified key cell populations, gene expression, and regulatory differences associated with the Jamaican fruit bat that also relate to human disease, particularly diabetes. So this doesn't seem to be a canonical model organism, kind of. So how did you get into?
Yes, I've always been, as a kid, fascinated with bats. I think they're amazing. They're like, my superheroes. Each one has their own superpower, like echolocation, and they all fly, and, blood sucking. Or, another really interesting thing is they can, they're mammals, but they can live on, just fruit or just nectar or blood. And so, in my mind, they've always been fascinating. And then, as I mentioned before, when we started the lab, we focused on limb malformations. And so I would go to these limb meetings and limb developmental meetings. And by chance, I happened to sit next to somebody at lunch, and Nicola Illing from South Africa, who actually can collect bat embryos in the wild. And so we started talking and came up with a really cool collaborative project where she collected bat embryos for us and then sent them to us in the mail. And then we did a lot of genomics on it, like RNA-seq and ChIP-seq and ATAC-seq on forelimbs versus hind limbs. And what's nice in terms of wing development in bats is that at a certain stage, the forelimbs and hind limbs look similar. But then suddenly the forelimbs, the digits start elongating. And also when we have our digits grow, we have webbing, and then we have cell death, and that's how we get our nice fingers. But in bats, that's actually suppressed, and that's part of the wing that generates a bat wing. And so we could do sort of paired comparisons where we got embryos at 3 different stages, the key stages of really wing development, and we did ChIP-seq, RNA-seq, and ATAC-seq on the forelimb, which sort of goes crazy, right, and becomes a wing, and on the hind limb that sort of stays normal, so to speak. And so we have a nice paired comparison. And we found a lot of cool regulatory elements important for what we think wing development. And then in terms of the diets, we've always been fascinated by this. And I was lucky enough to have a great graduate student, Wei Gordon, who's now a professor at Menlo College, who was very interested in diets. And, we were amazed by the differences in diets in bats. And as they're mammals, they're a great comparison to humans. And so we decided to look in particular on fruit bats because they can eat, 99% of their diet is fruit, which is filled with sugar, very limited in other sort of sources. And so, and they control their insulin levels beautifully and sugar levels. So you see like a peak and it goes down much better than insect bats. So we thought it would be really interesting also in terms of, better understanding for diabetes in a sense. And if you know the history also of GLP-1 with the Gila monster, where in the saliva, basically, that's where a much stronger, sort of more stable version of GLP-1 was found. You could envision also using bats, which are mammals, to try to find things that might be helpful in diabetes or other metabolic related issues. And so we were lucky enough to be able to go to Belize, which I'm going actually in 2 weeks also again to collect bats. And there's a lot of bats there, over 30 species of bats that we managed to collect. And a lot of them eat various diets, including, fruit eating, nectar eating, insect eating, and blood sucking bats, and even fish eating bats that have like crazy hind limbs. And we also collaborated, which was great to have. There's a lot of bat labs, in particular with COVID. A lot of people have been growing bats in the lab. So we were very lucky to collaborate with Tony Schutz in Colorado State University and Lisa Cooper in Ohio State that have colonies of bats. Tony has a fruit bat colony, and Lisa had an insect bat colony. So we could get samples from them and do this multiome to see if there's any differences in cell composition between fruit bats and insect bats, and also differences in gene expression and regulatory elements. And what we found was really fascinating, that there are a lot of differences. So, for example, if we look at the pancreas in the fruit bat, there's much more alpha cells and beta cells in the fruit bat that we looked at versus the insect bat that we looked at, suggesting, and in alpha and beta, there are of course glucagon and insulin, and so suggesting that they have more of these cells, and that's one of the ways where they control insulin and glucose levels much better than, for example, the insect bats.
So in the beginning, I mentioned that there are currently, I guess, 8 to 9 papers on bioRxiv, and that feeds maybe into my next question. So imagine you are due to submitting a grant proposal tomorrow, which you probably are. What would you have written into that? So maybe this can maybe be brought together.
Sure. I'd say from the bioRxiv, the one of them that I'm really excited about is, and we actually submitted and got recently a big grant from NCI to do National Cancer Institute, is a new cell therapy for cancer. And that's using basically adipocytes, fat cells. So, which I think is an under sort of utilized cell for therapy. So fat you can easily take out via liposuction in the clinic, and also implant via plastic surgery. It's highly used in plastic surgery. So there's everything there that works in the clinic, liposuction and, plastic surgery. So we thought, we could potentially play with fat in the middle and sort of modulate it, change it to do different things. And so what do you do with it? So one, you could potentially, so another nice thing I forgot to mention about fat is, we sort of have two types of fat. One is white fat, and that's throughout our body. And, if we have too much of it, we're obese, we're more prone to cancer, diabetes, and so forth. The other is brown fat that's up here near the neck. And that's actually sort of the good fat. It burns energy to heat. And as we age, it becomes less active. And, that's why a lot of us like to retire in warm places like Florida or so forth. what's nice about fat is you could take white fat, and if you upregulate at least 3 different genes, and each one individually can also do this, you can make white fat look much more like brown fat. So it's called beige fat, where it starts now burning energy and using a lot of glucose and fatty acids and so forth, and making it better. So we thought this could be a great therapy for obesity, for example, where you could take, white fat, make it brown fat, implant it back into the mouse, and, for example, put a mouse on a high-fat diet or use a obesity related mouse model, and it worked beautifully. And we're still sort of writing that out. That's not even published yet. But what we thought that could be great was also use it actually as a cancer therapy to outcompete cancer. And there was a beautiful work by a lab in Sweden, and published in 2022 in Nature, showing that if you put mouse models in cold, you increase their brown fat, you activate their brown fat, and that sort of outcompetes the tumor for reagents, glucose and fatty acids and so forth. And they also showed this on one patient and a few volunteers, just to show that they're upregulating. But when they did this, for example, on the volunteers, they were put in 12 degrees Celsius for 2 weeks with shorts and t-shirt for 5 to 7 hours. So it's not. And with the cancer patient that they did, they were a bit more conservative and so forth. But my concern with that, I mean, it's definitely doable, but cancer patients are frail and they're usually older. And so, as I mentioned, our brown fat becomes less active. And so it definitely could be a therapy, but we thought, if we could take out fat cells from a patient and modify them and put them back to outcompete, and that might be another approach that would be more therapeutically usable. And so we've done that in one of the bioarchives that we have out there, and it worked beautifully. Like, we were surprised initially. We thought it worked so well that we thought we messed up something in the cell lines or so forth. We had to repeat it a few times to believe that it really worked. And from the cell lines, we took it to a lot of mouse models, including xenografts and genetic mouse models, where, again, all of them have worked beautifully, suppressing the cancer. And we're also lucky enough to be able to work with Jennifer Rosenbluth here, who at UCSF, who's an MD PhD, who has a lot of mastectomies. And in the breast, there's a lot of fat. And so what we could do is take from the same patient their tumor and grow it. And then on top of that, we can grow from the same patient its fat that we modify and show that we can suppress the cancer in that patient using this approach. And finally, what we did there also was show that this is a universal approach. I mean, cancer is smart. After a certain time, it uses different metabolic pathways. And so we could show that, for example, we went after a pancreatic cancer, PDA, that uses uridine after a time when glucose is low. And so it starts using uridine constantly. So we took fat cells and upregulated a different gene called UPP1 that would make these fat cells start using uridine and outcompete for uridine with the cancer. And then we implant it in xenografts for these PDA, and we see that they outcompete and suppress that cancer also for uridine. Again, suggesting that this is a sort of a global way where you can use it. And we feel this is just the beginning. And sort of our grant now is really to develop these cells where we can upregulate many genes. We can have them respond to the tumor environment. We can have them secrete stuff. They're great secretors. So I really believe that this would be a really promising sort of new type of cell therapy for cancer.
Mm-hmm. So for the last 48 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think the most important thing is, that this is not me. It's all, I was fortunate enough to have amazing people in my lab that have done all this work, and I'm grateful for. And science is always a team sport in my mind, and work together. Lucky to have amazing collaborators also outside the lab. And I think the best things and the best ideas always start with, discussions and brainstorming and between people, and reading, keeping track of the science and the latest developments. And, just talking science is always a great way to get ideas.
And listening to podcasts.
And listening to science podcasts, of course. Yeah.
So thank you, Nadav, for your time and for being on the show.
Thanks so much for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.